Hello, welcome to our poster. My name is Ziji Zhang. I'm a PhD candidate in the Applied Mathematics and Statistics Department of Stony Brook University, under the supervision of Professor Yuefan Deng and Professor Peng Zhang. Thank you for your interest in our latest research and active learning workflow for 3D morphological analysis of bioimages. I'm happy to introduce my collaborators from Professor David Martus and Professor Benjamin Martin's biochemistry labs. I would like to present our poster in a few W's: what, why, and how. What is our problem? Why is it important? And how are we going to solve it? In the background section, we know that the invariant development and transparent body of the C. elegans enables a complete delineation of cell lineages throughout development and high-resolution microscopy. Here, we focus on a specific population of cells, the 6 myoblasts, which is SM cells. Several signaling pathways have been identified by classical methods, but the cell biological mechanisms underlying SM cell migration and pre-differentiation still remain largely unknown. To explore the cellular mechanisms, high-resolution and time-lapse microscopic images are often used to help analyze the behaviors of SM cells in vivo. These cells generate numerous actin-rich philopodia and exhibit complex cytoskeletal dynamics throughout their lifetime. And the accurate analysis of these biological phenomena with statistical robustness requires the interpretation of 3D images. Not only the inspection and quantification of such images are exceedingly laborious, but they are also extremely difficult for reconstructing 3D cells from meaningful 2D perspectives by manual annotation of subcellular geometries. Therefore, our active learning-based segmentation and reconstruction workflow is introduced to systematically measure and visualize the downstream cell 3D morphological outputs, and especially to interpret the morphological SM cell protrusions. The complete workflow are presented in this section. Raw images captured by confocal microscopy are pre-processed to augment the actual training set. Then, individual 2D images will go through a semi-unsupervised learning system for segmentation. This method, we have published it in another journal paper. The essential idea is that we train several agents networks which have different abilities to capture different features. Then, use a policy network to fuse the predictions and propose pseudo-labels. A reward network can evaluate if the pseudo-label is good or not, which could possibly propose additional annotation and retraining trials for the whole system. Then, the stacked prediction masks from this segmentation system will be input to a mesh generator based on Delaunay triangulation algorithm and reconstruct a 3D cell model. To evaluate our results, we compose a SM cell dataset with more than 1,000 fine annotations, and these metrics show that our workflow could outperform existing segmentation methods, including FIGImage. Regarding the reconstructed 3D cells, we analyze the cell sizes, cell shape topology, and deformations over time lapse. To further utilize the 3D measurements and explore the cell protrusions, we will detect specific motif shapes, their volumetric statistics, and track them over time. We are also working on applying this method to more applications, for example, the zebrafish tail bud. Our workflow significantly reduces time-consuming human workload and produces robust results validated by statistical analysis. We anticipate that our workflow and the ease of acquiring 3D morphological results will attract further image studies in the fields of computational biology, cell and developmental biology, and biomechanics. Please feel free to reach out to us if you have any questions. Thanks for your attention.